Dear everyone, I am Reedik Mägi, Professor of Bioinformatics from Estonian Genome Center, University of Tartu, from Estonia, and I would like to thank the organizers for an opportunity to present, and I hope you will learn something new today. My presentation will be about first overview of population structure in GWAS, as well as how to meta-analyze genome-wide association studies. I will also briefly talk about the transethnic meta-analysis. First of all, what is the population structure? Basically, if we take random individuals from any real population, they will not be unrelated individuals with exactly the same disease prevalences and the same allele frequencies. Populations contain individuals from discrete subpopulations, which often don't mix or don't mix as often. For example, different nationalities, or some individuals or families who have recently immigrated. And also, if there has been a recent mixture, that last or the new generation of mixed individuals have also different proportions of the genomes from different subpopulations, leading to different allele frequencies and then also different disease prevalences. And in the finite scale, there are also closely related individuals from the same families, which also creates additional structure into population data. For example, I have worked a lot on Estonian biobank data. I have some plots created based on it. And when we want to plot the population structure, what we usually do is we create a similarity matrix among individuals to find how closely they are related. Parents and children are 50% sharing genome, so are the siblings, grandparents and children are sharing 25%, and so on. Based on this, we can create a large similarity matrix among all individuals and use it to create principal components, which will describe the largest differences within the given dataset. For example, if we take a large proportion of Estonian biobank, 50,000 individuals, and look what are the first principal components showing, then we can differentiate between northern and southern Estonians, as well as western and eastern Estonians. It also helps to differentiate between nationalities. The blue dots in here are Estonians, green dots are Russians. And also, if we look into other nationalities, we can see that they also diverge from the Estonian nationality individuals. Therefore, there is a population stratification in Estonia. Interestingly, if we use a tiny proportion of our data, for example, we take 1,000 individuals and then create the principal components based on them, then if we have any very closely related individuals, like one parent-child pair, then these will hugely different from the rest of the population. In this case, the first principal component is created solely based on those two individuals who are very different from the rest of the population. If we take more individuals, like 50,000 individuals, then those nuclear families won't diverge and will be part of the main cloud. Thirdly, we have a plot where we have used Estonian data with a lot of different references. For example, in here we have created principal components based on all major ethnic groups. In here we have Europeans, Asians, and Africans. And if we plot Estonian data using those principal components, then we'll see that most of the Estonians seem to be Europeans, but there are some individuals who have Asian origin and then also some individuals who have African origin. So again, we either want to use that information in our analysis, or if we may want to remove some of the individuals from the analysis, just to be sure that we won't create wrong associations. Where does the population structure come from? It is mainly due to random drift of alleles in different populations, which is especially relevant if a population goes through a bottleneck, which means that at some time point, the population sample size has been very small. Let's say we have a parent population where individuals are carrying both blue and yellow alleles for a certain genetic variant. And let's say a small number of individuals move to previously uninhibited neighboring area. And by random chance, the individuals who are moving are carrying the blue allele. So as the number grows in the new place, we'll get another or new population where most of the individuals are carrying a blue allele. And we end up with two populations which have different allele frequencies. Now, we start a case control analysis and we pick individuals from both populations. If one of the populations have higher prevalence of disease, then we will find a false positive association from all the genetic variants that differ in allele frequencies between those two populations, including the example blue-yellow one. This is why traditionally human geneticists have been skeptical about case control design and prefer family-based studies. And here is an example of a problem. Let's say we pick our samples from two populations with equal sizes. First cohort has low blue allele frequency and very high disease prevalence. Second cohort has very high blue allele frequency and low disease prevalence. By random chance picking individuals, we will get most of our cases from the first subpopulation, most of our controls from the second population, leading us to the wrong impression that yellow allele is linked to higher risk of disease. If we would know that there are two populations, then we would analyze those two separately and we would avoid this false association. And here is an example how that problem exists in the real world. When we compare different ethnic groups, we see that there is a lot of diseases which have extremely different disease prevalences. In here, we have like 3 different cancers, breast, lung, and prostate cancer, and we can see that different ethnic groups have very different case or cancer prevalences for all those 3 subtypes. And now, if we pick a random genetic marker, let's say this microsatellite, which is highly varying among ethnicities with highly different allele frequencies, we'll find statistically significant associations between this variant and each of those diseases when running the analysis in combined dataset. One simple way to avoid it would be running matched analysis. It means that for each case from specific ethnic group, we'll find a control from the same ethnicity and then run some sort of pairwise analysis. However, this will not help in case we have admixed individuals. One example would be type 2 diabetes association analysis in Pima Indians. Each individual has different proportion of ancestors with European ancestry. As Europeans have higher risk of type 2 diabetes compared to Native Americans, cases tend to be individuals with larger proportion of European ancestry, and therefore the variants with higher frequency of Europeans seem all to be linked with type 2 diabetes. Another way to avoid this problem would be running analysis using family data. For example, family trios, where we compare children with disease with their healthy parents. One example of this type of analysis is called transmission disequilibrium test. But that analysis have its own problems. It's harder to collect family data compared to unrelated individuals. It is less powerful than case control design. And it is also very hard to collect parental DNA for late onset diseases like type 2 diabetes, where most of the individuals get disease in quite old age. So instead, we should try to use unrelated individuals and run genome-wide analysis and see if we can somehow correct for the population stratification. One way to adjust the association results would be using genomic control method. And in this figure, we have plotted p-values from genome-wide association analysis, which have been sorted so that the lowest p-values are here and the large p-values close to 1 are here. So if we take minus log 10 of a p-value, 2 means a p-value 0.1, 4 means p-value 0.001, and so on. And now we'll compare the observed p-values from our study to the expected one where the p-values are just totally random ones. And we would expect that most of the SNPs are actually not associated with a disease. And then in that case, they should align on this line. However, we see that they are all diverging, and then the p-values seem to be slightly smaller than expected by chance, telling that there might be some sort of population structure in our data. There are also some variants which are highly associated, but these are probably the true hits, and then there are actual genes which are associated with the disease. So what we want to do is to use a genomic control factor to push those p-values down to the line, making all the p-values slightly less significant. Another way to adjust those p-values would be using LD regression framework, which does more or less the same thing, but it also accounts for LD between variants. It means that it doesn't expect that all the variants in our analysis are uncorrelated and independent signals, but also uses information about the correlations between variants. Another very widely used option is to use principal components in the analysis, where you first throw out the ethnic outliers from your data and also use principal components as covariates in your regression model. When you add PCs from your data into your regression model, you can get rid of some of the population structure behind the largest differences in your data and get less biased effect estimates for your analysis. How it would work? For example, we could use the reference population like 1000 Genomes project data, which has samples from several major ethnic groups. And we'll then use a PCA analysis in that dataset to create axes and use the same axis in Estonian data to see how Estonian data maps compare to the reference populations. And in this plot, from in the left, we have colorful references, and Estonian samples are colored gray. And in the right plot, we have blue Estonian data, and all the references are gray. So we can see that out of our 200,000 samples, approximately a few hundred seem to be either East Asian or having African ancestry, and we should therefore be removed from the analysis. Next, we can use PCs calculated based on Estonian own data to investigate the population structure within Estonia. We can use the same PC analysis within external reference to see the final scale of population structure within Estonia. The first axis would distinguish between northern and southern Estonians and eastern and western Estonians, and individuals with different ethnicity would be on one side of the cloud, which had Estonians living closest to them geographically. Here we see Russians next to northern Estonians, as well as all the other nationalities diverging. If those axes were associated with a disease phenotype, then that fine structure within population could affect the association results and inflate the association p-values. To adjust for that, PCs are often used as covariates in the regression model of association analysis to remove that problem. As a conclusion, these multivariate techniques are computationally efficient method for adjusting population stratification in GWAS, and these principal component axes can affect large proportion of genetic variants and also help us to identify subpopulations of our data. We have to be careful when we are interpreting those principal components or eigenvectors. They, for example, may indicate extended regions of LD rather than population structure. One of these examples is inversion in the chromosome 17, which was shown in, or which is most prevalent in Icelandic data, and it actually creates a PC of its own, which is quite close to the first and major PCs or the most important principal components. The software tools to run that kind of analysis are usually Blink, Eigenstat, King, but there are many, many more software tools available. The state-of-art method for adjusting for population structure is actually mixed modeling, and it helps to adjust both for population structure as well for cryptic relatedness. And it considers the entire pairwise correlation matrix among individuals in the cohort. Here, for example, are some example QQ plots. First, for human height analysis, where 3 analyses were made. First, uncorrected analysis, where no population structure was taken into consider. That is the black line in here. And we can see again that all the observed p-values are much more significant than expected by random chance. Then we created 100 principal components and considered them in our regression model. That is a blue line in here. And we can see that that is much better, but still it diverges from the expected by random chance quite substantially. And third, we have used MMAX, which is one of the mixed model methods. And we can see that it gives us the best results, which is almost getting rid of all the population structure. In here, we have LDL cholesterol analysis, where we have 2 analyses. First, we have done, again, the normal association analysis using linear regression and not the mixed modeling. And in here, we have used MMAX, again, with mixed modeling. We can see that those high p-values or non-significant p-values create a cloud. But if we look into our top signals, then using mixed modeling is giving us slightly better p-values or slightly more significant p-values compared to running non-mixed modeling and afterwards using GC correction in our analysis. So mixed modeling results are close to uncorrelated results when there is a minimum evidence of inflation from genomic control, meaning that there is minimum population structure. Mixed model results are close to those corrected for principal components as the extent of inflation increases. It has advantage over genomic control of correcting population structure of it because it corrects each SNP individually and independently. And it can be applied to binary traits using logistic mixed modeling, although some care is needed when the interpretation of results is occurring. Mainly the effect sizes are not the same. And currently it is a method of choice when analyzing large biobanks data. And software tools are Bolt LMM, Saige and Regini. These are some mainly used software tools. As a summary, population structure can lead to spurious associations if disease prevalence and allele frequencies vary between subpopulations. We can use information from markers scattered across the genome to test for the presence of population structure, and then try to use either GC, inflation factor, principal components, or mixed modeling to create read of population structure and cryptic relatedness. The second part of my lecture is about meta-analysis. Why it is important. One way to boost the power of our analysis is to collect more and more samples to detect rarer variants with modest effects to the disease. And currently, largest biobanks contain already hundreds of thousands or even millions of participants, but even that may not be sufficient to analyze very rare disease, or we will lose a lot of power by just analyzing one cohort. Alternatively, we can combine those GWAS results of several biobanks. And for that, we only need to exchange the summary statistics of variants, and there is no need to share individual level data, which is often difficult due to ethical and legal obstacles. One of the most commonly used methods for combining association results of several cohorts is fixed effect inverse variance weighted meta-analysis. In here, we're basically calculating the weighted average of effect size then this is weighted by inverse of variance, which basically means that the smaller the standard error or the confidence interval is for an effect, the more that cohort contributes towards the real effect or the meta-analysis effect size. And if you have very large cohort, in that case, you will get very good estimate for your effect, and then it has very narrow standard error, and it means that larger cohorts or larger populations will have a larger contribution in that weighted analysis. And then we'll get that effect, and we can test for the association over all studies, getting a p-value for our meta-analysis result. And commonly used software tools for it are, for example, Metal and GWAS. There is an alternative for the analysis in case your effect sizes are not directly comparable. It's called Stouffer's method, and in that case, you will not use effect size with its standard error or odds ratio with its confidence interval, but you are rather using p-values from your results and sample size n to the calculation. You first calculate the z-score from your p-value, and then you will again weight it by the n, and you will combine those statistics from across your populations to get the meta-analysis p-value. These fixed effect meta-analysis assume that the real effect of a variant is the same in all contributing cohorts. We can test it if it seems to be true using a test of heterogeneity. One of them is Cochrane test, or Cochrane Q test, which checks how much each cohort effect size is deviating from the average. And then there is an alternative matrix I-square, which can be also calculated, and it shows probability that there is a heterogeneity in effect sizes. For example, I-square 0.5 tells that there is a 50-50 chance that there actually is some heterogeneity in the signal. And if there appears to be an effect of heterogeneity in your association signal, it is important to figure out where it comes from. It can be due to some hidden difference in your cohorts, for example, some containing old and some young people, or it can be due to mislabeled effect allele in one of the cohorts. So the effect is in opposite direction. And here is an example of fixed effect meta-analysis with heterogeneity estimate. This is called forest plot. So each cohort result is for one variant, which we are looking in here. And this red rectangle shows the effect estimate from that cohort, and the whiskers show the confidence interval. And if we look, we can see that, for example, in here, those two cohorts have opposite effect directions in here as well. But most of the cohorts seem to have the same effect direction. And if we meta-analyze them all together, we can see that there is an effect which is larger than, odds ratio is larger than 1. And if we look into the heterogeneity among those cohorts, we can see that we can basically draw a line through most of the cohorts, telling that there is very little heterogeneity. Q is 12.12 with p-value 0.15, which is statistically not significant, and the I-square is 35%, telling that there probably is no heterogeneity and everything is fine. But if there is an heterogeneity, we can try to use random effect meta-analysis. And in that case, we don't assume that the variant in all cohorts has the same fixed effect, but rather that the effect has a distribution. It means that some of the cohorts variant has larger effect to the disease, and in some cohorts the variant has smaller effect. And altogether, the cohorts should have like a normal distribution of effect sizes. It has a bit more complicated calculation formula where we first estimate the random effect estimate like this, and then we use that estimate when we are calculating the weights for all the cohorts. So we just don't take the inverse of variance, but we now add that random effect variance component into the weight. As I said, sometimes this random effect meta-analysis is used, but it would be very important to investigate where that heterogeneity is coming from. Is it due to population? Is it some sort of interaction with exposure, some environmental risk factor, or which was not adjusted in the association analysis, or is it some sort of error and wrongly labeled alleles? And for fixed or random effect analysis, there are several software tools. Meta can do fixed effect meta-analysis, Gamma can do both fixed and random effect meta-analysis, and Metasoft actually has its own test for random effect model, which is a bit more efficient version of the random effect model. And finally, if the cohorts which we want to meta-analyze have different ethnic backgrounds, we cannot expect that the effect sizes are comparable. It can be due to different environmental factors or different LD patterns in a genetic region under observation. And random effect model is not the best option in that case, as different ethnicities. We expect that cohorts which are more similar to each other have more similar effect estimates, and we want to consider that information in our meta-analysis. One of the methods to use for transethnic meta-analysis is meta-regression. We first create a similarity matrix of all cohorts based on their variant allele frequencies. For example, in here, we see that the first few PCs very well differentiate between cohorts from European, African, American, and other ethnicities. And we have implemented a meta-regression model into Mr. Mega software, which then uses those PCs and expects that similar cohorts have more similar effect sizes and is able to test for association even if the effect estimates vary a lot between ethnicities. So, as a summary of meta-analysis, meta-analysis of GWAS summary statistics allow to increase the power to detect associations because we can put together the information from many biobanks. If we are running our analysis in a single ethnic group, for example, in Europeans, the most commonly used method is fixed effect inverse of variance weighted meta-analysis, and it assumes homogeneous allelic effect across all the studies. So it is important to assess the evidence for heterogeneity. And then there is software designed for GWAS meta-analysis with features that allow the strand alignment and SNP filtering and everything, so you don't have to do it manually. And it is also very important to model that heterogeneity in your analysis to see if there seems to be heterogeneity in your analysis. And in case of transethnic analysis, you would like to use other methods like meta-regression, for example. I would like to thank you for listening. And if you want to learn more about those topics, then please register yourself into the next summer course held hopefully in July. Thank you.